Thank you for having us here. I think it's really nice to come all the way. I know it's very hot. It's much hotter than Pune, but I'm very happy to be here. This is such an envious facility which you have in this building, and also when I learned the kind of microscope that is being demoed here, it is really very special. So hopefully I can give you one application of that microscope that we have standardized recently. And actually, before I go into my talk, can I know what is the composition of the audience, in the sense that I know that you are teachers, so you probably know theory very well, maybe better than me even. And but are there biology teachers, or are there other subjects, physics?
Yes, students as well, PhD students.
And PhD students as well. So are there people from the physics background? Or sorry, what did you say? Some people who are physicists. Yeah, so then I will like to, say one thing, which is that you will know a lot of the optic theory. And so then this might be more of a biology centered talk, but then I'm a biologist, I'm not a physicist. And since this talk also attempts to think of microscopy beyond optical resolution, really one of the key expertise, I think, comes from the side of physics and from the side of chemistry, and hopefully I can convince you that. And biologists, of course, gain a lot from these kind of discoveries, right? So I'll try to summarize some of the principles, but maybe the in-depth physical principles may not be part of this presentation at all. Okay, so what I'm going to do is I'm going to combine talking about super resolution and also live imaging. So live imaging is something, like Richa said, is something which we do a lot in the lab, and we have specialized in not only imaging in vivo sort of large samples. So we are interested in looking at Drosophila embryos developing, so these are large samples, and we specialize in looking at processes while these cells divide at a high resolution, and then trying to understand what kind of dynamics takes place in these embryos, and maybe some of that will also come out through this talk. So what I thought I could do, since I'm not a physicist and I'm not a chemist, is tell you a little bit of what I understand from super resolution, what are its goals, and what have we as a personal experience from there. So one of the things, I just outlined a few goals for this talk, which is that why do we need super resolution, right? So electron microscopy does exist. Electron microscopy gives unprecedented resolution for biological structures. But one of the things which it does is that these are fixed samples. It's not so immediately easy to label each component using electron microscopy. Suppose we wanted to have multiple components labeled, and we wanted to look at their organization in a cell, that becomes hard. And then if we wanted to look at things live, electron microscopy is certainly not the way. So there has been a huge wave of discoveries by scientists to basically improve optical resolution, so that we can look at labeled molecules inside living cells and even fixed cells, right? So fluorescence, which, since you have gone through a lecture on confocal microscopy, you know that fluorescence is used very predominantly in looking at distribution of proteins inside cells. And can we improve the resolution of the confocal microscope or of optical microscopes in general to try and localize molecules with unprecedented precision, right? So with increased precision. So that involves breaking the optical barrier, right? So we have to think of improving this resolution. And what I'm going to do is I'll give you a brief summary of very few sort of famous and upcoming super resolution methods, and practically just 3 of them. There are more than these which are being worked on as we speak. And I'll give you an example of STED and how we have used it to image a certain process in embryo development. And I'll give you an example of how we have used photoactivation to image embryos live, and then the use of photoactivation in one super resolution microscopy, which we've not done, but that is something which it led to, and hopefully will lead to. Right, so since you have had a lecture on confocal microscopy, you know that in general there is, okay, I can use this as a mouse. Is there a pointer? It's okay, you can see this, right? So in general, the light microscope resolves and the resolution limit comes to around 200 nanometers. Now this is something which depends upon the refractive index of the material, it depends upon the objective lens, it depends upon the problem that light is a wave, alright? It's not a straight line, but it's a wave, and because it is dependent on the fact that there is a lens there and the light is a wave, based on both of these constraints, there is a resolution for optical microscopy which is at around 200 nanometers, depending upon how good the objective is actually. Electron microscope, of course, can go a lot more, and the goal for super resolution is in this region, where it is trying to improve the resolution better than 200. Okay. So let's look at a quick revision of confocal microscopy, and since this microscope which you're going to see also has a confocal application. Essentially, confocal microscopy allows us to image a particular region inside the sample, and the way this does it is because of the presence of the pinhole, right? So the pinhole allows for light to be acquired from only a particular focal plane within the sample and cut out light which will, for example, be incident on these parts of the pinhole, which is coming from other focal planes of the sample, right? So confocal microscopy is very dependent on the concept of the pinhole. If we open the pinhole, then we will get light from many different regions of the sample, and depending upon the application that we want and depending upon the amount of light that is present in the sample, we might choose to open or close the pinhole. So if we are at really optimal pinhole closure, right, then we can to some extent get the resolution which was documented in the previous slide of around 200 to 250 nanometers. And what does that mean, right? So what that means is that if two particles are 200 nanometers apart, then the microscope can distinguish them as separate particles, alright? And if they are closer to each other, which I'll show you in the next slide, then we can't distinguish between them, okay? And one more concept to remember here is that since we are using a point source of light, so typically lasers are being used. But since light is a wave, then it is going to flow through the objective, and it will either sum up and increase the number of photons. So basically, you will have either interference or the lack of it, right? So basically, if you have because of interference of light, because light is a wave and not a straight line, this point is diffraction limited. It is diffraction limited also because of the kind of lens that we can put over there, right? So because of the refractive index of the particle, right? So I'm not going into the theory of this. So this is where I wanted to say that physicists will know this a lot more in detail. Now, Ernst Abbe came up with a way of finding out the resolution, and again showing the same thing in slightly different terms, is that if these two spots are away from each other and they are about 200 nanometers away from each other, then we can actually separate them. What you can also see is that when the waves actually come together with each other and they sum up, then in the center you will see a very bright spot, and here you have destructive interference where you start seeing these dark regions which are formed, and then you see these various different regions which are present, which then, so basically every particular bead, right? So imagine this as a bead which is which is let's say 100 nanometers in size, everything which is less than 200 nanometers in size is still going to appear as 200 or 250 nanometers in size depending upon the resolution of the microscope, right? So then what happens is that if these beads are let's say close to each other, then the objects will interfere and you have some sort of cloud which is formed here. And if they are very close to each other, then what you see is that essentially you just get one big blob, right? So you don't know when there is a highly intense signal, whether that intense signal is coming from one bead or is it coming from multiple beads. And the goal for super resolution microscopes is to be able to resolve these peaks better. Okay, so can we, for example, I mean we know, for example, let me just give an example of GFP, right? So GFP is about 5 nanometers in size. If you think of, you know, how many GFPs can fit in this 200 nanometers, huge numbers, right? Hundreds, let's say, can fit in this 200 nanometers depending on which kind of three-dimensional orientation it is. So now we are not able to localize this better than that 200. Right? So if we are looking for, let's say, placement of a couple of different particles in a particular region of the cell. So let's say, for example, is the endoplasmic reticulum, and we have 2 or 3 many different kinds of proteins which are present there. Then they are basically going to just appear that they are in the general same space, but which set of molecules are close to each other is not something which we can decipher using this kind of microscopy. So the goal for super resolution is to improve the resolution such that you can go from this general appearance of a blob-like structure to more distinct visualization of individual molecules. And then the task for super resolution is such that you want to improve the resolution approximately 20 times that of a confocal microscope. And in practice, this means that when 2 fluorophores are separated by around 10 nanometers, they should appear as individual particles, right? And by conventional microscopy, they are always going to be together, and they will appear as, unless they are more than 200 nanometers apart. Okay, so that is the goal for super resolution microscopy. Now, there are a few methods which have gained popularity in the last 2 decades, I would say, and of them I just very briefly introduce structured illumination microscopy and go a little more in detail with an example of our own experience with STED. I show you something more about photoactivated light microscopy, localization microscopy, and only compare photoactivated localization microscopy with stochastic optical reconstruction microscopy. They are very similar. Okay, these two are very similar to each other. This one and this one in some sense are similar to each other, but we'll see how they are achieving super resolution. So one thing to note, which is the difference between STED, which is right here, and PALM and STORM, which is what they are called as, is that in the case of STED, there is a goal to just be able to capture photons from a very, very small region of interest. Alright? So now we saw that, we saw the very disk. We basically saw the region that is being shown by these particular peaks. Right? So it has this major peak and then it has these minor peaks. Can we, for example, reduce the airy disk itself? Right? So can we reduce the spread of this light itself? Can we capture light from really a very small region? Right? So that is a goal of these first two, especially STED, alright, not so much structured illumination. And the goal for PALM and STORM is, can we highlight or activate only one molecule at a time? Okay, so both ways we can achieve super resolution. Because if we highlight only one molecule at a time, then we know that that one molecule is likely to be very small, and we can computationally know its position and place it in the brightest portion in the middle of the place where the light is coming out. Alright, so we'll see how this looks like with actual examples. Now, structured illumination microscopy, I'm I'm going to quickly go through this, basically uses a set of grids. It uses patterns where what it is trying to do, I'll go to this example. So for example, let's say that this is the sample. It's not a very good idea of a sample, but let's say that this is the sample. Okay, good. So now let's say that this is sample. It's a boring sample actually to put, because even the illumination is in a patterned way. Now it can be in various different orientations, and when this orientation of illumination overlaps with a sample, which can be complicated, okay, so this is the highly simplified example which is shown here. When these two overlap with each other, it will give interesting patterns which come up, which then are called as Moir patterns. Now these are the ones which basically allow for sharpening again of the position from which the photons are coming out. Alright, so after getting these patterns, there is a computational exercise which is done by the microscope, which then allows for improving the resolution. Alright, so basically one great thing, and watch out for this method. I'm not going into details for this method, but watch out for this method. It may be a very simple method which will actually allow us to look at living samples at a very high resolution. Alright, and something may sort of surpass even the other super resolution methods which are present in today's age. So in general, one would, for example, prepare the sample. It can either be a live sample or it can be a fixed sample. We are actually imaging the sample and then putting all of these grids on the sample, and then looking at interference pattern, and then actually doing an image analysis to localize all of the signals in a more precise manner to give rise to improved resolution. Okay, so there's something to watch out for the future. This is an example of a blurry image which is seen just by widefield fluorescence, and then you can see the localization of these structures much better. This is actually a neuromuscular junction where each of these positions is a point at which a synapse is formed between a neuron and muscle. And you can see how nicely through structured illumination microscopy this has become better. Okay, so now coming to STED, and that is something which is there here, invented by Stefan Hell, a physicist who was able to basically improve the limit of detection. And how did he do it? So one of the ways in which he did it was, okay, the principle again for biologists hopefully, and physicists will anyways understand this better, is that we want to get light only from that very small central region. Alright, so which means that I want to deplete light from all other regions around. Okay, so if I get light only from a very small region, then I can be sure that that is actually coming from a point which is now getting highly resolved, right? How do we do this, right? So there are two beams in the Leica cell microscope. So one is the general spot for excitation. So let's say if it is something like a green fluorescent molecule, then it's going to be excited by the 488 nanometer laser, and we use a beam. So for example, one could use the 592 nanometer beam, and this beam will actually give rise to converting all the molecules on the side in a dark state. Right? So because all the molecules around the central zone are in a dark state, we can precisely localize the center. Alright, so this is a donut beam. Now one can adjust the size of the donut. You can imagine that if the donut is big, then you will get many more pixels which are getting excited in the middle. If the donut is really small, you want to make it as small as possible so that you get light only from one molecule. Okay, so there are problems with this as well. That one molecule has to be very bright. Okay, so we have to think of tags, good tags which can do this. We have to use those fluorophores which can turn into the dark state, right? So they have to be able to be depleted by all the depletion lasers which exist in the system, right? So then there is, of course, huge amount of information of which kind of fluorophores one could use. So for example, now one can change a view of a blurry sample to sharper localization of the same set of molecules. Now, what I'm going to show you is our personal example of an application through this. And like I said, we study Drosophila embryos, and our interest is in understanding how cells remodel in these embryos. And what you're looking at here is a point at which there are multiple cells which are actually present at the periphery. So these are multiple nuclei which are present at the periphery. The embryo is syncytial, which means that multiple nuclei are present in common cytoplasm. But if we look at each of these individually, then what we can see is that around each nucleus there are these convolutions which are present of the plasma membrane above. And these are villi. Right? So villi are present in a lot of different kinds of cells. They increase the surface area of the plasma membrane, and they allow for communication between the cells more effectively with the outside environment. Now, what we also see is that this is a stage at which the numbers of nuclei are multiplying again and again, and they are increasing in number. So they are going through the cell cycle or syncytial division cycles, and you have a stage where there is a cycling taking place between interphase and metaphase. And what we know is that this is through very old experiments, through scanning EM experiments. It is known that these villi-like structures are present at the top, and then they are not present in metaphase. Alright, so they expand and they go away. So now, can we visualize this with markers which label actin better? Right. So this is also to show you the kind of shape remodeling which I am talking about. When you look at each syncytial division cycle, you can see that there is a stereotypic shape change from circular organization of cells to polygonal organization of cells. And what you see here, when the thing is a circle, then you see more ruffled presence of the membrane. Right, so it's ruffled. That's all you can make out with a confocal microscope. So watch the point at which it is a circle. You can see that the membrane is ruffled. Most likely that ruffling is actually coming about because of the presence of villi. Alright, so can we visualize these villi better? And this is something for which we use STED. Now, a lot of times for STED you can't use a DNA dye, so we try to use other dyes which could mark DNA. So here we are, for example, using phosphoristone 3. This also works nicely for us because in interface, for example, phosphoristone 3 does not label the nuclei. It starts labeling the nuclei in prophase, and you can see that in metaphase it labels the chromosomes beautifully. The chromosomes are present at the metaphase state. Okay. So it also not only allows for labeling of the DNA, but also it allows us to decipher the stage of the syncytial division cycle in these particular embryos. We can label the membrane with a marker which is membrane specific. In this case, we want to look at villi, and these villi are rich with actin. So we are using an actin marker, right? So you can see that this actin marker is somewhat changing its distribution. It may be a little more ruffled here as compared to here, right? But now can we visualize villi better, right? So what we did was we used STED, and these are images from the same STED microscope. We looked at different division cycles, division cycle 10, 11, 12, 13, and we are looking at interphase and metaphase. And what you can hopefully appreciate over here is that we can see these villi-like structures. They are abundant. They are present around each individual cell. They are present in these cycles very clearly, whereas in this particular cycle now you can't see them very well. Right? You can also see that especially in nuclear cycle 11 and nuclear cycle 12, you do not see them in metaphase. You only see them in interphase. Right? So they are getting remodeled most likely from interphase to metaphase stage. And you can see that in cycle 10 also maybe there are more villi over here and less villi over here. But then one would like to count this, right? One would like to count that there is a remodeling which is actually taking place. So this is we've just filled this particular cap with a yellow color to mark the boundaries of a plasma membrane which surrounds one individual nucleus. we can look at this, and for our sake we had to figure out which cycle it is, right? So just by figuring out the area of each cell, we can now say that okay, if the area is around 250 micron square, then it is in cycle 10 and so on, right? So these kind of quantitations allow you to document better the stage of development that the system is in. Now let's see what the difference between confocal and STED is, right? So that microscope has both, and really in that microscope, if you have the 100x objective, which has an area of something like more than 1.4, 1.5 or something, that itself is also great. It's absolutely great. So you can make out these villi-like structures very nicely even in the confocal, and you can make them out slightly better in the STED. How do we know that it's better, right? So what we can do is we can look at what is the improvement in resolution we are getting. You can clearly see this is sharper as compared to this. So let's just focus on the interface stage for now. In the metaphase anyways, there are fewer villi, and then we can look at what is the spread of the signal that we are seeing for each of that villi structure. And what we see is that the spread of the signal now is much sharper for the STED, which is calculated by this full width half maxima plot of the fluorescence intensity, right? So you can see that the spread is smaller for the STED as compared to the confocal microscope, right? And this is both in the case of interface as well as in the case of metaphase. Now one more thing on top of this which is done in that microscope is that one can put deconvolution algorithm on top of this, and hopefully Ashish will explain to you more about this at the microscope. This again uses the concepts of point spread function for the system itself, right? How much can the system resolve? And then it is a mathematical improvement of this image, and I'll show you the amount of improvement that we get again by drawing a line around it. What we see is that in interface you have a smaller full width half maxima spread of the intensity as compared to the STED itself.
Making this at the STED confocal, like how you?
Yes. So we take the STED image, and there is a way of doing it at the microscope itself, right? So that is something which will improve the resolution even more. In fact, it's recommended for most applications that we take the STED image and then run it through deconvolution.
Got it.
Yes. Okay, so then we want to know something more quantitative about the system. So what we've done is in ImageJ, ImageJ is a free software there where you can now begin to start counting these structures that can be clearly seen. So we've just labeled them with yellow for you to see how many structures, for example, are there in this one particular stage like interface. And in metaphase now the structures actually reduce quite a bit. So we can come up with two sorts of calculations. We can come up with an obvious calculation which is that of microvilli number. And here you see that there is a decrease in number in metaphase as compared to interface. And we can come up with a quantitation of microvilli length, which then is also decreased in metaphase as compared to interface. So this then, then you can start speculating about what could be the mechanisms which are leading to this remodeling between these two stages of the cell division cycle, right? And then it hopefully, come up with some molecules which do this. So, just to summarize what we have, we think STED is good for, and also some of literature supports it as well, is that it can be, first of all, successfully used for many different kinds of biological samples, and that's one of the reasons why it has gained popularity, especially in places which use a variety of samples, not just cell culture, right? Because cell culture, a lot of cell culture is really flat cells. But when I'm thinking of embryos or maybe other tissue samples, it's very attractive to try out STED. It can allow for improved resolution and increased depths inside the sample, and this could be almost up to 30 microns inside. You will get better resolution. It of course does need a choice of fluorophores which can go into the dark scatter, right? So we want to be able to get light from very small regions in the sample. So the fluorophore has to be such that the depletion laser can convert them into the dark state. But there's a huge set of fluorophores documented to be good for this. So that information is available. There is very often, if you're a biologist and you're already doing immunostaining experiments, then you will have slides which already have fluorophores which work very well for STEM. Labeling of DNA sometimes can be a challenge. I mean, we can't have hex in the samples. Sometimes it interferes with some of the samples, and hopefully Ashish can tell you more about this at the microscope. So then one may want to use some other kind of label to actually label DNA if we want to know the stage of development for in that particular tissue at that time. Okay. So in summary, what you've seen now, this is through a hardware-based solution. The hardware has a depletion laser which is converting the molecules into a dark state and allowing for improving resolution. So it definitely, hopefully convinces you that if I illuminate a few, then that will improve resolution, right? So that hopefully convinces that point. Now, super resolution actually very quickly got a Nobel Prize after the work of these three scientists, and Stefan Hell is who I spoke about, and the STED microscope is what you just saw previously. But both Moyna and Petzig worked in a manner to think of the fact that if I am able to just highlight single molecules at a time, then I know that anyways because of the point spread function or because of the airy disk concept, it is going to be a blur which is around 200 nanometers. The center of that will be where the particle is placed. So one can use a localization method. If I, let's say, highlight one molecule at a time, one can use a localization method to computationally place the position of the particle, right? So even that can be done. And Moyna essentially looked at the physical principles of this even in the 80s. It was really long time back when he said that this should be possible and it can be possible in a variety of different ways. Petzig, who was working on this also for a long time, is the one that pioneered the palm system, and I'll hopefully give you some flavor of how he came to it, right? So essentially, what is the palm system? So this is photoactivated localization microscopy. So always try to contrast this to what you're going to see instead. It's to me somewhat easier to understand than the STED principle, because what you're doing is you are actually highlighting particles sequentially, okay? So you are highlighting them somehow. One can think of many different ways of highlighting them. One way is photo activation. So maybe the tag could be photo activated, and I'll give you one example of how photo activation can actually be used beautifully in live imaging as well in the next few slides. But come back to its real use in super resolution. So one can activate single molecules, let's say. Once we activate single molecules, we know that that's how the light pattern is going to show. There's going to be interference and so on, which will show this interesting diffraction pattern. And where is the molecule? The molecule is most likely placed in the center, right? So one could computationally localize the molecule after acquiring the light from each of these individual patterns, right? So what is one doing? We are activating few molecules, and I'll give an example of that. Then we are bleaching them because now we know where they are placed. So we are activating, we are localizing, then we bleach them. Okay, we hide them again, and then we activate, localize, bleach. And if you do this repeatedly again and again on a biological sample, then you can imagine that you can start seeing structures which are actually coming up in the sample, right? So in the beginning, you will just have one or two molecules. Then you will have more molecules, and you can do this iteratively again and again and again. And then you can actually look to see how that sample is, and in that sample, how these individual molecules are placed. So this now is giving you unprecedented resolution in nanometers, okay? And one can typically look at a number of molecules adjacent to each other in a space, in somewhat a three-dimensional space. So to begin with, this method was actually used such that it was used only for surfaces of cells. So one could only do it in the top 5 or 6 microns easily. But this has become better over the years, and one can look at more depths, right? Okay, so the main task for this, and you will obviously already realize, is the development of photoactivatable probes, right? So we want a probe which we can now attach to the molecules inside the cell, which we can activate one at a time, right? That's the only way we can actually activate them sequentially. And what I'll do is, I will first tell you something about the development of this photoactivation tool. It's a wonderful tool just for you in live imaging. So since it's something that you will think of over and above super resolution, use this as an example, and then we look at its use in photoactivation, photoactivated localization microscope. So one knows that with the advent of GFP and tagging of GFP to proteins, one could begin to look at protein dynamics inside the cell, right? So one can do, for example, these different methods like FRAP, flip, and photoactivation. So just briefly to see what's happening in FRAP. If let's say the protein of your interest is a cytoplasmic protein, so this is a documentation of a mammalian cell where the cytoplasmic protein is labeled with GFP and is present inside the cytoplasm, you can see a uniform label of the GFP present there. Now, if you want to know how fast the molecule is exchanging in the cytoplasm, then what you could do is a method called fluorescence recovery after photobleaching, where we are actually photobleaching a particular region of interest and then allowing for recovery of the fluorescence in that region. So the fluorescence here has gone down to 0. Now you are allowing for recovery to take place, and then you are looking to see where the recovery is coming from, right? So imagine that this particular label is in a very precise location, let's say mitochondria or the endoplasmic reticulum. Then what you will see is that the label is constrained in a particular location. The recovery will only come from a particular region inside the cell, let's say endoplasmic reticulum, and only the endoplasmic reticulum signal will start getting diluted, right? So FRAP is an excellent way of knowing that molecules are being exchanged in this entire cell, and also the location from where the molecules are being exchanged, right? So even dynamics tells you a lot about how compartments are connected to each other, right? Flip is basically the opposite method, where instead of bleaching once, you keep continuously bleaching. So what is happening here is that you shine a high intensity laser, it turns dark, then you shine it again, it turns dark again. So one of the steps which is missing in this particular schematic is that you're waiting in the middle, right? So there will be molecules which come in from this region into this region, then you're shining another high intensity laser and bleaching them, right? So if you do this again and again and again, essentially you are depleting the entire system, because it is slowly passing through this fluorescence sink, right? It is like switching off. The molecules which come in this zone just switch off, okay? So the rate at which the molecules come in the zone will now start getting highlighted by this depletion curve which is shown here. And one of the methods which is exactly opposite to FRAP is photoactivation, right? So this was developed by George Patterson in the Jennifer Lippin-Gottschalk's lab. And what he did was, he basically improvised on a particular facet of GFP, which is that under a certain set of conditions, this is actually the wild type GFP, which is found in jellyfish, had the ability to undergo photoactivation. Alright, so what do I mean by photoactivation? So if, for example, we take a UV laser, we shine it on the wild type GFP in a particular location, then it suddenly gains fluorescence. Alright, and then what we can do is a pulse chase experiment, not in a gel, which is what routinely speaks of pulse chase experiments, but inside the cell, right? So in the cell, what we can do is we can now look to see where will these molecules which are activated go, right? So now here, for example, we switched off the fluorescence and we are asking if other molecules from this region are coming in. here we are now activating this region and asking where will these molecules go, right? Are they confined to a certain place? And so it allows you to do pulse chase experiments inside the cell. And I cannot, complete this talk without showing Jennifer and George. So I had the very nice luck of being a postdoc in Jennifer's lab. This was at the time just after when George had invented the photoactivatable GFP, and this was a very nice time to see the use of photoactivation come up in various different facets of cell biology. George, an extremely talented biomolecule developer. We don't have George is no more actually. He passed away very recently of cancer, but was an absolutely wonderful individual and a wonderful scientist, a very talented scientist who developed better photoactivated probes of many kinds. Right, there is a PA Cherry out there, which is fine-tuned by him to work better for many different applications, and there's a PA GFP out there, which is again fine-tuned to all of these photoactivation tools for many different applications. So they had developed this tool, all right, and remember that Moiner. So I'm going to shuttle between, photoactivation principles for super resolution and just live imaging back and forth. Moiner had already, through his work, shown that if I can highlight a few set of molecules, then I can localize them, okay, and improve resolution. Bedzik had also done this, but Bedzik at that point did not know how to activate just a few molecules in a system. Okay, and then he basically you must hear actually the Nobel talk of all these people. It is fascinating, and especially for Bedzik, it is dramatic. It is almost like a dramatic Hindi movie. You must watch it for what discovery can happen when you're just looking for that one solution for that one problem. Okay, and then there's ups and downs. So there were times in which he realized that no, we can't do it. Okay, there's no way of doing it. And then what ended up happening is that he said that okay, I'm going to go and do something else. Till he said, let me go back to reading and find what the biologists have done, and then discovered that photoactivation has been found. So now can we couple photoactivation with super resolution? Okay. So anyways, first I'll show you one application of photoactivation which we have done, and hopefully you appreciate how it can allow you to look at very crowded systems. So one of the other interests we have is in studying mitochondria and embryogenesis, right? So mitochondria are this powerhouse of the cell. They are organelles which allow for giving ATP for these different cells and so on. They are maternally inherited, so if there are any maternal diseases in mitochondria, then it's very important that embryo development, aspects of embryo development are studied so that we can know that if embryo development does not work correctly because of mitochondria, what are those different steps of development which are affected, right? So we are using Drosophila as a model system because it's a very powerful model system to understand what's happening with distribution of mitochondria. Now, if some of you have seen mitochondria in cell culture, you know there are these worm-like structures which are present all over cells, very nicely distinguishable organelles in cell culture. When we first looked at embryos, we were quite disappointed actually, to say the least, because this is what mitochondria look like. They're really small. They're around 300 nanometers in size, and they are huge numbers, like abundant, millions of mitochondria present in the Drosophila embryo. And it's very difficult to see their movement, let alone their change in shape, right? So mitochondria also change in shape, and it's very difficult to actually visualize any of that. So one of the ways we could ask if mitochondria are localized to a particular place was by using photoactivation. So we made this mitochondria photoactivate mitophoto PAGFP, and then what we can do is activate mitochondria in a localized region and ask how they are getting moving in one place or another. And in this case, actually, what you see is that they are pretty stationary. They are present in one place. When I, for example, photoactivate a particular region with mitochondria, they actually just stay there, right? And a non-activated region doesn't show increase in intensity at all. What else can you do? In this crowded system, you can activate just a portion of the mitochondria and ask whether they are getting translocated in different directions. Right? So mitochondria could move down or up, and you could ask in what direction are they moving. And when we, for example, just looked at mitochondria by staining for mitochondria, what we saw was that in early cell formation, so there is a process of cell formation which these embryos go through, there is a lot of mitochondria inside the embryo, alright? This is an internal plane. So if you look at this, essentially it is a section which is taken from the top of the embryo inside. And what you see is that there's abundance of mitochondria in the inner planes at around 15 microns. And towards late cellularization, you start seeing that there is increased mitochondria now in more apical planes, alright? So mitochondria most likely during the formation of these cells, which is a process called cellularization, are migrating from down to up, right? So just looking at the fluorescence distribution and quantifying the fluorescence distribution allows you to predict that there is some such movement which is taking place. And then one can do an interesting photoactivation experiment with mito-PHFP, where we can photoactivate a region below the cells, and what one sees over time is that there is an accumulation of fluorescence now on the top, right? So all the fluorescence over here now is on the top, which means that there is this translocation event which is taking place. So photoactivation as a tool is a fantastic tool to look at to follow a signal live and to see where it is going, right? So what we saw was that the region where the mitochondria photoactivated, which was the base of the cells, was then losing fluorescence, and the apical region was gaining fluorescence, and the only way it can gain fluorescence is because there is a movement of that particular signal from down to up, right? So now coming to super resolution. This allows you to, if you use very low intensity of laser light, activate very few molecules at a time, alright? So depending upon the amount of fluorescence which is present in each of the molecules, you will see a different brightness for each of those spots, alright? So there is still a task for the field to actually become better and better at labeling each of those molecules, to become better and better to evolve the chemistry of these molecules, like instead, for example, to have a high contrast between the dark state and the active state. In this particular case is to have a high contrast so that you have enough photons which are coming out from each of the molecules, right? So this is so that you can then localize them very precisely, right? So you can see a nice spread of the light, and then you can, for each of these molecules, actually localize them, which is pretty much the center of the position where the light comes from. And then over time, when you keep doing this again and again, so again to remind you, the way this is being done is that you photoactivate or activate the molecules, you localize them computationally, alright, and then you bleach them. So these are now no longer again viewed. And then you repeatedly do this, and then finally you start seeing the presence of the full structure. And this is the improved resolution which you get. For example, this is the confocal image, and this is a super resolution image for cytoskeletal structures in this particular sample. Storm is a very, very similar method, also something which I showed you amongst one of the 4 methods, where instead of photoactivation, it is just using other chemistry of the fluorophores to basically switch them on at a time, and then again localize them. Alright, so it's using the same method where in palm we are switching them on. Here it is using other chemistry of the fluorophores and is trying to localize them. So this is something which is sold by Zeiss, and this is something which is sold by Nikon. And both methods are also very attractive to start looking at the localization of molecules at a high resolution. So just in thinking about the 2 methods, at the end I thought I'll just bring these 2 slides. These are a part of Stefan Hell's Nobel lecture actually. And so what they are trying to do, these methods, is essentially look at 2 states, an off state and an on state, right? And then And then, so instead, it is trying to actually come up with an on-state in a very, very precise region. In the case of palm, it is trying to come up with an on-state of few molecules. Instead, since this is being done sequentially in the sample, and hopefully you notice that when you're looking at the demo sessions, you don't have a problem of localization. It is, the light is coming from wherever it is placed there. Whereas in this case, the activation is sort of taking place in a stochastic manner. So you have to do this again and again till you have saturation of activation in a particular sample, because that's the way of localizing, right? And then again to reiterate that in both of these methods, you're looking at the distinguishing feature of an on-state and an off-state. And in the case of palm, because you can very easily, the premise itself is by looking at single molecules getting activated, you are automatically getting single molecule resolution, right? So whereas in this case, it just depends upon how well we can achieve that task. So the resolution of this system is slightly more than this system depending upon the sample. But let me remind you that STED is great for a variety of different samples, right? So if one wants improved resolution and we have complicated biological samples, which we often do, then STED is a great method of choice. Okay, so that just brings me to an ending summary of this talk, where what I've shown you hopefully is the importance of super resolution. Remember, we had electron microscopy already existing, but super resolution aims to identify the distribution and also dynamics, right? So something which I have not shown in this presentation, but more and more has been a goal for STED as well as for PALM and STORM, is to try and look at molecules of those single or molecular dynamics of those single molecules which are being highlighted across these living cells, in addition to just fixed cells. And also for fixed cells, it's great because I can go up to 3 to 4 tags, and then I can coordinately localize them at a high resolution, right? So in that way, super resolution microscopy has achieved a lot of success. So when you think about this, it's a physics problem, and it is trying to solve interests of biologists. But finally, the concept of localization has already come about, all right? So that we can localize molecules is there. So it is now up to the chemists, and a lot of chemists are interested here, to start developing more stable, more intense, and maybe even smaller size dyes, which then can be used to mark these different proteins, right? So it is a chemistry problem at the end of it. So I think we will see a lot of improvement in the future for better dyes for both of these methods. I only spoke about dyes which you can put in the dark state, but think of all the chemistry which has similar molecules which can sort of exist in two kinds of states, right? So if molecules can exist in two kinds of states, and if there is a state which exists at a very small fraction, then we can use that particular molecular state to invent an imaging method, right? And let's say use that in the STED mode. Then we can essentially use that to a super resolution method, right? So essentially identifying different molecular states by knowing the chemistry of the molecules will improve the detection and the localization possibility for the use of all of these molecules in super resolution, right? So these three sort of take-home messages come from studying a little bit the super resolution system, and I'll just leave you with acknowledgements from my lab of people who standardized the visualization of actin villi, that's Devasmita, Amrita and Gayatri, over time. And Sairi worked on making the photoactivatable meadow GFP and the funding, and Jennifer and George have always to be acknowledged for inventing the photoactivatable GFP in their lab. Thank you.
I'm Dr. Ajay Shani. I'm SPA over here. It was a nice talk, ma'am. You have presented it well, and I just got all more information about that. I just wanted to know a bit information about super resolution. Like as you have told, like we can see the dynamics of the molecules too in the living cells.
Yes.
So
yes.
like instead, as I know, we have to fix the cells or fix the particular.
No, you can use live cells. So look at. I will recommend you to actually see Stefan Hell's Nobel lecture, which is 2014 lecture, right? So which is still old, I would say, in terms of just development of this method. So what he has done is he has actually looked at the remodeling of synapses from a live mouse brain, and you can see the remodeling of those synapses in a live mouse brain really well also. So yes, it is true that there is a depletion of the signal, and at the end of it, it does depend upon how intense the light emission from each of those states is. But definitely try out live systems. There would be something or the other, some or the other diane system which will work. So if you look at the literature, there are examples of places where it has been used for laboratory.
You have done it for laboratory?
No, we have not done it.
So at your system also you have you
We want to do it. We just
100x.
haven't done it.
Yeah, but you can do it 100x only, right? Like
So we want to do it at 100x. Yes. Yeah. For us, that is good actually.
Yeah, I mean for 100x. I mean if you go lower resolution, then
Yeah, but this is the general range. There are also some more interesting ones which are present in the range of 300, 350, but and those we can't see with our eye. The far red, the ones which also emit in the far red, we can't see with our eye, but then optics can be developed to see them and digitally, process them on the computer.
Can you explain the dark color?
The dark field, right? So so basically what that is doing, and hopefully again Ashish can explain to you better about this because I'm a biologist, just keep that in mind. So I'm going to go back to this principle, and I think unfortunately my computer is not here, but there are better diagrams for illustrating this. So what's happening here is that typically you will have an excitation spot which then gives rise to fluorescence such that it gives rise to a big diffraction limited spot, right? So can we, for example, just excite very less, right? And then can we excite fewer molecules as well as also excite very less? The way we can do that is by converting all of these molecules on the side in a dark state, and this is done by the depletion laser. So for each fluorophore, it turns out you will have a redshifted specific depletion fluorescence which will allow them to remain in the dark state. Okay, so they are present in the active state and they are present in the dark state. Also, one more thing which I have not done here is I think we should look at the Jagromsky diagram to actually understand this better. Okay, so that is also something I can share with you. I can actually share this entire set of things with you if you want to refer to this, but really it's given very well in some of the other lectures which are given by Stefan Hell itself. So you should look at those lectures.
please go ahead.
I had one biological question, like the localization of apical versus basal microlocalization. So
Yes.
could you attribute any significance to this, like cellular?
Yeah, so I mean this is a methods talk and I would love to give you a biology talk because I really was hoping I'd talk about research more than methods, and I mean I'm a research scientist, okay, methods has come along on the way. So this is a wonderful study which we did, which were papers in 2020, so you should take a look at it. We were interested in trying to find out whether there is mitochondrial change in distribution as the cells form. Alright, so this is a point at which cells are forming, and we saw then just by looking at the fluorescence in this manner that there is a change in distribution of mitochondria which is taking place, right? So first they are abundant in basal sections, and then mitochondria become more abundant in apical sections, right? So there is a translocation of mitochondria which is taking place. So what we did then next after this was actually look at microtubule motor mutants, and we showed that, for example, when we have dynein mutants, then mitochondria remain in these basal sections, and when we have kinesin mutants, then they actually translocate to apical sections even earlier. Okay, so there is a motor control of this transport which is taking place.
As if the mitochondria have moved even above the nuclei now, right?
Yes, they have moved above. They are exactly above the nuclei. So in fact, they form this very interesting cloud of mitochondria. And I mean, one of the nice things is we are now imaging all kinds of organelles at this point. So this cloud is a very interesting mitochondrial cloud which is present around ER. So this
So
is
mitochondria ER interaction.
it may be. So it's very attractive to think of that question in this particular context. So one is the motor control. The other is that, why are they doing this, right? So now these are really tall cells which form at the end. They are like almost 40 microns tall. And one of the reasons one could argue that this translocation of mitochondria is important is so that mitochondria are present in every location to provide local energy or function. And what we found was that when we use, for example, mutants of DRP, so DRP is this dynamic protein, which is responsible for fission of mitochondria, we have these long, big mitochondria which are now clustered at the bottom. Okay, they don't move to the top. And also there is a change in activity of these mitochondria. So what we see is that this closing off of cells is aberrant in this particular system then, if the mitochondria are big and they are sort of dysfunctional in their activity. So that is something which we could show in this particular study. What is that mitochondrial function which leads to an aberration in these big fused mitochondria was actually a decreased production of reactive oxygen species. So it turns out that for this particular system, there is an appropriate concentration of reactive oxygen species which needs to be present so that this process of cell elongation takes place appropriately. And when there is reduced reactive oxygen species which are actually present, then we are now seeing the entire process go down. So then the question comes about what is reactive oxygen species interacting with, and I mean I can go on, and there's something really interesting out there, but I can talk to you separately also.
More questions? So if not, I thank Richa
Thank you.
for this really nice talk.
Okay.